Proteins are the engines that keep life running. I am Mark from Mentivia. Welcome to an in-depth look at proteins, the incredibly diverse and essential macromolecules that drive almost every process in living organisms. From catalyzing reactions to building structures and transporting molecules, proteins are the true workhorses of the cell. But how do these complex molecules come to be? And how does their intricate shape dictate their vital functions? Today, we'll explore the fascinating journey from simple building blocks to a fully functional protein. We'll examine how amino acids link together in precise sequences, how these chains fold into three-dimensional structures, and how that final shape determines what the protein can do. Whether it's speeding up chemical reactions, forming cellular scaffolds, or moving substances across membranes, each protein's role is encoded in its structure. Understanding this journey is key to grasping how life operates at the molecular level. The fundamental building blocks of all proteins are amino acids. There are 20 common types of amino acids, and each one shares a basic structure: a central carbon atom, an amino group, a carboxyl group, and a hydrogen atom. What makes each amino acid unique, however, is its side chain, or R group. This R group can vary greatly in size, shape, and chemical properties. It can be polar, nonpolar, acidic, or basic. And these differences are crucial for how the final protein will fold and function. Amino acids link together in a process called dehydration synthesis, also known as a condensation reaction. During this reaction, the carboxyl group of one amino acid reacts with the amino group of another, releasing a molecule of water and forming a strong covalent bond called a peptide bond. As more amino acids are added one by one, a long, unbranched chain forms. This chain is known as a polypeptide. The sequence in which these amino acids are linked is incredibly precise and is determined by genetic information. The first level of protein structure is the primary structure. This is simply the unique linear sequence of amino acids in the polypeptide chain. Think of it like the specific order of letters in a very long word. This sequence is determined by the genetic code within the DNA and is absolutely fundamental. Even a single change in this sequence can have profound effects on the protein's overall structure and, consequently, its ability to function correctly. Once the primary structure is established, the polypeptide chain begins to fold into local, repeating patterns, forming the secondary structure. These patterns are primarily stabilized by hydrogen bonds that form between the backbone atoms, specifically the carbonyl oxygen of one amino acid and the amino hydrogen of another. The two most common secondary structures are the alpha helix, a coiled shape resembling a spring, and the beta-pleated sheet, which is a folded zigzag pattern. These structures add stability and rigidity to specific regions of the protein. The tertiary structure is the overall three-dimensional shape of a single polypeptide chain. This intricate folding is driven by interactions between the R groups, or side chains, of the amino acids. These interactions include hydrophobic interactions, where nonpolar side chains cluster together away from water, hydrogen bonds between polar side chains, ionic bonds between oppositely charged side chains, and strong covalent disulfide bridges which form between two cysteine amino acids. This unique 3D conformation is absolutely critical, as it creates the active sites for enzymes or binding sites for other molecules, directly enabling the protein's biological function. Some proteins are composed of two or more polypeptide chains, or subunits, that associate together to form a functional complex. This arrangement is known as the quaternary structure. These subunits are held together by the same types of interactions found in tertiary structure, such as hydrogen bonds, ionic bonds, and hydrophobic interactions. A classic example is hemoglobin, the protein in red blood cells that transports oxygen, which consists of four polypeptide subunits working in concert. This level of structure allows for even more complex functions and sophisticated regulation. The precise three-dimensional structure of a protein is paramount to its function. If a protein loses its specific shape, a process called denaturation, it also loses its biological activity. Denaturation can be caused by changes in temperature, pH, or exposure to certain chemicals. This highlights why maintaining stable internal conditions is so vital for living organisms. It ensures that proteins retain their correct shapes and can perform their essential roles. Imagine you're building a protein yourself. You start by selecting individual amino acids and dragging them into a specific linear sequence. As you link them, forming peptide bonds, you're creating the primary structure. Then, as the chain grows, it spontaneously begins to fold into alpha helices and beta-pleated sheets, forming its secondary structure. Finally, the R groups interact, pulling and pushing the chain into its unique, functional three-dimensional tertiary structure. If multiple chains are involved, they then assemble into a quaternary structure. This entire process is a marvel of molecular self-assembly. In summary, proteins are built from amino acids, linked by peptide bonds to form a polypeptide chain primary structure. This chain then folds into local patterns, secondary structure, and in an overall 3D shape, tertiary structure due to R-group interactions. Some proteins further assemble into multi-subunit complexes, quaternary structure. This precise folding is absolutely critical, as a protein's specific three-dimensional shape directly dictates its biological function, making it an indispensable component of life. Thanks for watching. If you enjoyed this video and want to keep expanding your knowledge, make sure to hit that subscribe button. It's the easiest way to stay connected and never miss new content. For even more free educational resources, head over to mentivia.com, your hub for videos, e-learnings, and articles across science, history, and beyond.